Okay, right. Next chapter, synapses, excretory products and their elimination. So, excretion is the elimination of metabolic wastes like ammonia, urea and uric acid from the tissues. The process of excretion, elimination of metabolic waste like ammonia, urea and uric acid, all are nitrogen compounds from the tissues. And types of excretion: ammonotelism, ureotelism and uricotelism. So, ammonotelism, removal of ammonia, and ureotelism, removal of urea, and uricotelism, removal of uric acid, right. So, ammonotelic, the process of excretion of NH3 urea, the process of excretion of urea, the process of excretion of ammonia, the process of excretion of urea and the process of excretion of uric acid. And ammonotelic animals, examples, most important, most very important. So, ammonotelic animals examples: aquatic invertebrates and aquatic insects, bonifacious and aquatic amphibians. Most of the aquatic organisms are ammonotelic because which one is highly toxic? Ammonia is the highly toxic. Excretion needs excess of water, so that only most of the aquatic organism releases ammonia. And NH3 is readily soluble in water and is excreted by the diffusion through the blood surface or gills in fishes as ammonium ions. Kidneys do not play any significant role in removing of ammonia, right. Through the body surface, ammonia is released in the aquatic organisms. And second least toxic, that is urea. And examples: cartilaginous fishes, terrestrial and semi-aquatic amphibians, frogs, toads, and aquatic or semi-aquatic reptiles, alligators, turtles and mammals, or releases urea. In liver, NH3 is converted into less toxic urea, so it needs only moderate quantity of water for excretion. Ammonia most require water releasing, means water is required for releasing of ammonia, and urea so moderately required. Uricotelism, very less water is required. Some amount of urea may be retained in kidney matrix some of the animals to maintain desired osmolarity, right. And not completely, urea is not released for maintenance of osmolarity in the body. And uricotelism, and here it is water insoluble and very less toxic material. High toxic ammonia, moderate urea and very less toxic uric acid, and water is no need for excretion. And examples: insects, uricoteric organism examples: insects, some land crustaceans and land snails, and terrestrial reptiles and birds. Ureotelism and uricotelism are needed for water conservation, right. And some of the excretory organs in animals: so protonephridia, flame cells, inware, in platyhelminthes, in platforms, rotifers, and some annelids and cephalochordates also presented. And protonephridia are primarily for osmoregulation and excretion also, as well as nephridia in annelids. Annelida in phylum Annelida consists with nephridia, helps in the removal of nitrogen wastages and osmoregulation. Earlier we discussed in earthworm. And Malpighian tubules in insects, Malpighian tubules are excretory organisms, helps in removal of nitrogen wastages and osmoregulation. And antennal or green gland, this also in arthropods, so that is aquatic. In crustaceans, prawns, crabs, those are all. And kidneys in higher organisms, all are excretes through the kidneys. And human excretory system, it includes kidneys, ureters, urinary bladder and urethra. What are the components of human excretory system? Kidneys, ureters, urinary bladder and urethra, right. So, structure of kidney: reddish brown bean shaped structure situated between the levels of last thoracic and third lumbar vertebra. Location of kidney is most important. Between levels of last thoracic and third lumbar vertebra, right. So, length of the kidney 10 to 12 centimeters, width of kidney 5 to 7 centimeters, thickness 2 to 3 centimeters, average weight 130 to 170 grams, right. And it enclosed in tough 3-layered fibrous renal capsule. Once again, go through this diagram and labeling also in NCERT. On the concave side of kidney, there is an opening, hilum or hilus, through which blood vessels, nerves and lymphatic ducts and ureter enters into the kidney. So, this is called hilum. And in this region, so all the blood vessels are enters and nerves and lymphatic ducts and ureter is enters. Hilum leads to funnel shaped cavity is called renal pelvis. This is renal pelvis. Kidney and labeling is the most important. So all the labelings are most important. So this is called renal pelvis, and renal pelvis have the projections are called calyces. And a kidney has outer cortex and inner medulla. This is called cortex, and inner region is called medulla. And the medulla region consists with medullary pyramids, right, and projecting into the calyces. These medullary pyramids are presented on where? On the region of calyces. Cortex extended in between the medullary pyramids and renal columns. So this is called renal columns. So this region is cortex only. This is extended into medulla. This is called renal columns, are also called as columns of Bettini. Each kidney has nearly 1 million tubular nephrons. There is a structural and functional units of kidneys are called nephrons. How many approximately presented? 1 million, right? So next one, nephrons are structural and functional units of kidney, and each nephron has majorly two parts, glomerulus and a renal tubule. And this region is called glomerulus, and remaining part is called renal tubule part. So glomerulus, a tuft of capillaries formed by efferent arteriole and fine branch of renal artery, and blood from glomerulus is carried away by efferent arteriole. Efferent arterioles and forms blood capillaries, tuft of blood capillaries, the bunch of blood capillaries, and formed as glomerulus. And here and emerged out as in the form of efferent arteriole, right? And renal tubule, it begins with a double walled cup like Bowman's capsule. So here renal tubule parts, so there is Bowman capsule, PCT, and LOH, and DCT, collecting duct. Here given a cup like Bowman capsule which encloses the glomerulus. So glomerulus is enclosed by the Bowman's capsule, and glomerulus plus Bowman capsule is called Malpighian body. Only this region, glomerulus and Bowman capsule, is called Malpighian body, are also called renal body. And the tubule continues with a PCT, a proximal convoluted tube, Henry's loop, and distal convoluted tube, and a collecting duct. Proximal convoluted tube and LOH, DCT, distal convoluted tube, and LOH, and proximal convoluted tube, PCT, and LOH. This is LOH, and this one is distal convoluted tube and collecting duct. Proximal convoluted tube. So proximal in earlier region, proximal, and convoluted, convolutions are presented, tube, and distal end region, distal convoluted tube, right? In between that U shaped, that is LOH. Descending, this is descending limb of loop of Henle, and this is ascending limb of loop of Henle, and finally the DCT opens into collecting duct, right? And next one, the DCT of many nephrons opens into the collecting duct, and collecting duct extends from cortex to inner parts of medulla, and each nephron do not have single collecting duct. The group of nephrons opens into collecting duct, one collecting duct. They converge open into the renal pelvis through the medullary pyramids and in the calyces. All the collecting ducts directly converge into the opens into the region of medullary pyramids in the region of calyces. Malpighian body or renal corpuscle, PCT and DCT are situated in the renal cortex. Loop of Henle dips into the medulla. Up to here, all the regions are presented of the nephrons presented in the cortex, and remaining this portion is presented in the region of medulla, right? Okay, that is important. And next one, the efferent arterioles from the fine capillary networks, peritubular capillaries around the renal tubules. A minute vessel of this network runs parallel to the Henry's loop, forming the U-shaped vasa recta. So this is so from the efferent arterioles, the network of the blood capillaries along with the length of the nephron, so that is called peritubular capillaries. Earlier it is called peritubular capillaries, and runs through the LOH U-shaped. This is called vasa recta, which are majorly helpful for maintenance of osmolarity in the kidneys, right? And types of nephrons, majorly there is two different types of the nephrons are present in the kidneys, cortical nephrons and juxtamedullary nephrons. So which are more? Cortical nephrons 85%, cortical nephrons, juxtamedullary 15%. In this, Henle's loop is very short and extends only very little into the region of medulla. Cortical, the cortex region only. So whenever Henle's loop is very short and extends only very little into the region of medulla. So vasa recta is absent or highly reduced in cortical nephrons. So what about juxtamedullary?